Yeah, so what I want to do in the next 30 or so minutes is give a bit of background as to how we, from the finding of one of the most active stem cells of the mammalian body that you see here in action in the gut, how that took us totally by chance into developing organoid technology, starting from adult, so from primary tissues rather than from iPS cells or ES cells. And then I'll give a few examples and also some recent work that we have tried to use organoids for various applications that take us closer to patients and closer to the clinic. I should also stress that indeed I'm currently a Roche employee, member of the executive board, but what I'm talking about is actually the work from my lab in Utrecht, which I still supervise from a little bit of a distance. So what you just saw was the stem cells in the gut in action, sit in the bottom of the crypt. They produce massive amounts of daughter cells every day. Nick Barker in the lab found this new marker LGR5 that then helped us to find the stem cells of the gut. We now know it probably marks all epithelial stem cells of a mammalian body when they are active, not when they're quiescent. So they divide every day in a mouse, and that then inspired Toshio Sato, now a professor in Tokyo, to see if we could actually take one of these stem cells then out of a mouse, but since then we've all translated this to humans and to patients. Can we take one stem cell and recreate the growth conditions in a crypt where they normally reside, the crypts of Lieberkühn, to encourage them to divide? And the intention was to make a lump of stem cells, but that's not what we got. You see actually the three recombinant proteins that we use for mouse intestinal stem cells, Wnt activator, our spondin, turned out to be the ligand of our marker a little bit later, epithelial growth factor, and a BMP inhibitor, no serum. Still, we do this in Matrigel, so in 3D, inspired by Mina Bissell. And Matrigel is still magic. I don't think we have anything yet that can replace Matrigel. Anyway, so a single stem cell grows out, makes these structures rather than just lumps of cells. And when Toshi started looking carefully at what these really represent, there was a central lumen. The central lumen is lined by all of the mature cell types of the gut epithelium. We probably now recognize about 12 to 14 different cell types that together help us to digest food and take up nutrients efficiently while protecting us against what is inside our gut lumens. And these buds that you're sticking out turn out to be sort of equivalents of crypts with multiple stem cells. So we start from one, but we make many of these organoids, and then daughter cells that divide and move to the center of this lumen here. Several very unexpected observations. One was, first of all, that they built these structures at all in a very reproducible manner. Second, that they actually don't stop doing this. So these stem cells grew essentially forever. We now have them in culture for more than 10 years, and still every week they grow about tenfold, creating these structures, which we then called mini guts. We now call them intestinal organoids. And as I said, they contain all of the cell types of the gut epithelium. I'll discuss one of our recent projects where we exploit this. And even they put the different cell types in the right positions of these structures. Now, they don't really look like guts, but when we line them up by growing them in bundled collagen gels rather than in Matrigel, which is a more amorphous gel, you can see that they fuse. You see it in the top here. But then when we start from one stem cell, we have enough of these organoids lined up. And this was done by Norman Sachs, now active in California. Actually, we can make these long tubes that really start looking like real guts. So a central lumen, that's where all the differentiated cells, when they're about 5-6 days old, they actually undergo apoptosis, they end up in the lumen, like what happens in a real gut. And then we have multiple crypts that line this central lumen of the gut tube. And in this epithelium, we essentially see all of the cell types. Now, one immediate way of using this, an example would be to not only grow normal cells, but also grow cancer cells. We just heard about cancer in patients. And we can do this side by side. So that was then quite unique. So in the lab, we would have a sample of normal gut epithelium. You just saw a patient with colon cancer. On the right, we would have the tumor tissue growing. And this would now allow us, first of all, to read the DNA of the tumor, but that could have been done directly on the primary tissue sample. But most importantly, we have the tumor cells in a petri dish growing probably forever, representing the tumor of an individual patient. And this can now be screened these tumor organoids, and in theory then we can predict, possibly by screening cancer organoids, what would be a good drug or drug combination or treatment regimen for an individual cancer patient. Now, this turns out to work well. We were not the first. We were working hard on this, but then scooped by a beautiful study in Science 2018 that shows that if you do this carefully in well-controlled phase 1 and 2 trials, the predictive value of organoids, so basically the organoids are grown from patients that are already participating in trials. The organoids are exposed to the same treatment regimen that the patients will experience in a trial, and then we can compare the outcomes. Did the organoids predict well, or did they not predict well what would happen to the patient? And it turns out that at least in this first study, 85 to 90% correct predictions came out of just screening organoids. And then the next 4 papers, we had some involvement, but these were all clinical groups that we helped a little bit. And now there's probably about 30 or 40 papers that all show that if you do this carefully, it's not very simple yet, but you actually can very well predict for classical chemotherapeutics, for radiation, and for targeted therapeutics. You can predict if a patient will not respond with very high fidelity. What this particular version of the test doesn't show is whether immune checkpoint inhibitors, for instance, will work, because we have no immune cells in this particular version. There is actually now a number of papers where they are added, and they also score very well. And angiogenesis inhibitors will, of course, also not score, because these tumors, as they grow in the lab, are not dependent on a blood circulation. So just rapidly, how we now actually use these organoids. We made a small biobank, there's a number of years ago, from colorectal cancer patients, either the hypermutated, the MSI version, or the MSS, the non-hypermutated. It's about 25 or so here. We now have biobanks of hundreds of these patients, and we think we can really capture the diversity of each and every individual malignancy by creating these large biobanks, living biobanks. In this particular initial biobank, there was one patient who did not have any of the known mutations in the EGF receptor KRAS pathway, and that was patient 18. All other patients would have a hit, an activation hit in this pathway. So we asked, how can we do something with it? With CRISPR, it's actually very easy to make modifications in organoids. We use this extensively, and I'll give more examples a little bit later. But what Hayana Ross in my lab, together with the Hugo Snippert lab, did is they now engineered into this one tumor sample here that did not appear to have an effect in this pathway. They engineered in the G12D mutation in KRAS, very common in colon cancer. And then they exposed, so you see the original organoids of this particular patient that are highly sensitive to a drug combination of an EGF receptor inhibitor and a MEK inhibitor. You see how in a very short while, all of the cells that are happily growing at the start of the experiment are dead. You see that the nuclei are now crumbling. If we do the same experiment with this very same cancer, but now we added a single base change, there is one letter in the DNA code is changed, activating KRAS. So now this cancer has an active EGF receptor KRAS pathway, and this makes this particular organoid entirely resistant to the very same drug combination that was happily killing the cancer cells when they were still in their original state. So this basically this was a predicted outcome, but based on this now but many other labs are using the patient organoids, so tumor organoids directly coming from a patient that you can actually track back to the patient to learn about drug sensitivities, drug resistances, but also maybe to help develop new drugs for these cancer patients. This is one of the 2 stories this one is actually not submitted yet, but I want to show you what is feasible in organoids. What you see here is human small intestinal organoids, or healthy organoids. Kui G and Lin last year published a protocol that really optimized the growth of small intestinal organoids. And in practice, what we do, we need a tiny biopsy, or we need a little bit of a surgical resection, but no more than a cubic millimeter. It would suffice, but it needs to be alive, should not have been thrown in formaldehyde by the pathologist, which often happens. But if we get it alive, we put it in Matrigel. Actually, we basically turn it into small fragments, 10, 20 cells, put it in Matrigel, add the growth factors that for every tissue have to be optimized, but there are always variations on a theme that I just showed. And then they start growing. And here what Guiwei and Lin did is they knocked in fluorescent proteins in 3 different marker genes. MUC2, mucin-2 marks goblet cells. So in bright green, you see there's lots of goblet cells. They're also quite common in the gut epithelium, so that's fine. Defensin A5 marks Paneth cells. They typically sit at the base of the crypt. You actually see these dark cells here. This would be the crypt equivalent. This is the central lumen. So there are Paneth cells there, and they're orange. And then cells that have received a lot of attention recently, the enteroendocrine cells of the gut. So there's about 6 cell types. They all are marked by this purple marker here, chromogranin A, in this knock-in. So you see the purple cells. So one of the 6 cell types, I won't talk about them more, but is the L cell, and the L cell makes this magical GLP-1 hormone that is now becoming rapidly very popular as an obesity drug. So what these cells do, they essentially measure what passes by in the gut. They secrete their hormones and then instruct the owner of that gut to start eating or to start burning calories, etc., etc. And GLP-1 is one of the about 20 hormones that these enteroendocrine cells make. But I won't talk about this anymore later in this talk. So to give a little bit of background to this study, about 7 or 8 years ago, together with 2 scientists in Utrecht in Holland, Florijn Dekkers and Jeff Beekman, we developed a test for cystic fibrosis based on a personalized test for individual patients. Now, all CF patients have a mutant CFTR gene. The mutations can be in different parts of the gene. Of course, there was a drug developed by Vertex that helped about half of the patients, the most common form of CF, but many other patients had a rare mutation. And that was the reason to see if you could set up a personalized medicine test. And what the CFTR gene encodes is a chloride channel, which can be closed and open. And when it's open, it allows chloride ions to pass from the top of the cell into the lumen of the organoids. And when that happens, water is also transported to the lumen of the organoids. And this takes care that at the apical domain, the mucin layer, the slime layer, the mucus layer that sits on top of the apical domain, so inside the gut, inside the lungs, inside the liver, etc., that mucus basically doesn't become very viscous, doesn't become very sticky. And if you don't have this channel, you're a cystic fibrosis patient, you cannot transport water into the lumen, and you get this extremely viscous mucus, and that gets you in trouble. Now, the assay, I won't talk about CF anymore, but the assay is simple. Here you see rectal organoids. So we take small biopsies from children. It's a painless maneuver. In about a week, we have these organoids growing. When they are really grown, we turn them green, and we can open the CFTR channel by adding forskolin, which raises cyclic AMP, or cholera toxin, which actually naturally opens the CFTR channel. And when you do that, chloride ions will flow into the lumen of the organoids, and at the same time water will flow in, and that's why these organoids so rapidly swell. So when they do swell, we know there is a functional CFTR channel. It also shows that cholera toxin induces water transport into the lumen of the gut, and actually what it does in a real patient, it causes a massive watery diarrhea, and that gets important a little bit later. Now, this is the normal response. If we now take a sample of tissue from a cystic fibrosis patient, we now have a biobank of about 800 of these patients. You can see there's very little lumen at the start in these organoids, and when we now add forskolin or cholera toxin, they don't really swell at all because they don't have the channel, the chloride channel, so they cannot transport chloride ions and they cannot transport water into the lumen. This is, by the way, the reason why CF carriers are probably highly resistant to cholera toxin, and also why CFTR mutations are so common amongst Caucasians, because medieval times in Europe, there were many cholera epidemics, and if you would be a carrier of mutations, you would not develop, this is the mini diarrhea, but you would not develop diarrhea, and you would actually survive more easily a cholera infection. Now, if you take these organoids here and we now expose them to the Vertex drug or Cambi, wait for a few hours, and now give them cholera toxin or forskolin, the channel is restored by this magical Cambi drug, and now we restore the swelling assay. So this is a functional assay for the CFTR channel in cystic fibrosis, but also in normals. So keep that in mind. Now, the story is basically entirely done by Daizong Wang, a Chinese postdoc in the lab. And he noted that there's many single-cell mRNA studies being published on a variety of different tissues in human and mice. And actually, when you do this on gut epithelium of human and mouse, it turns out that there is a cell type, a novel cell type that humans have, but mice don't have. It's called the BEST4 cell. It expresses a gene called BEST4, but it also expresses a number of other genes that I'll show you. And there are many studies that picked up this cell type. It looks very much like an enterocyte, the most common cell type. It's actually related to an enterocyte and had not been noticed before this fantastic new technology was developed. So, but this cell was only known as a gene signature. Nobody has cell lines, and in mice, because they don't exist, you cannot really study them. So what Daizong set out is, so can I see if it can create these BEST4 cells in organoids? So these are the genes that are expressed by BEST4 cells in humans. CFTR is very prominently expressed. It should be somewhere here. Here it is. BEST4 itself is here as another channel protein. There are many channel proteins. The BEST4 cells are predicted to help balance ions like chloride, sodium, potassium across the apical membrane, and also water and pH, so control pH. You also note that SPIB is a transcription factor that appears to be unique to BEST4 cells, and that also gets to be important a little bit later. So this is the BEST4 cell. So on its apical domain, it has CFTR, already showed it, so it allows chloride ions, and then water follows. So it basically keeps water on the outside, on the luminal side of the epithelium. Also important for the story is yet another molecule that sits on the apical side, GUCI2C. It is a receptor that, when triggered by its ligands, you see the ligands here, the GUCAs, will create cyclic GMP, and cyclic GMP, like cyclic AMP, will actually open the CFTR channel. This is how heat-stable enterotoxin from E. coli causes a massive diarrhea. It presumably binds to GUCA2C, and then through cyclic GMP opens the CFTR channel. So cholera causes diarrhea through the use of CFTR, the activation of CFTR, but also this heat-stable enterotoxin, E. coli, will do the exact same thing, albeit by a slightly different mechanism. So do we have BEST4 cells in the organoids? So can we actually do experiments on these cells rather than look at mRNA the signatures and predict what they do? It turns out they are there. What Daizong did, he knocked in TD tomato, red fluorescent protein, into one of these marker genes of this novel cell type, CA7, and indeed, lo and behold, they are there. They're red cells, and when we now stain the same organoid for the BEST4 protein. So this is a knock-in done with CRISPR. This is staining afterwards, and indeed everything that is red here is BEST4 positive. So we believe that this is really a good indicator organoid line. On facts, we can see them. They're pretty rare. It's 1.4% in the steady state of these organoids. So we've actually learned that the intestinal epithelium is pretty active in terms of immune responses or controlling immune responses, and many of the interleukins, now that we can study them in vitro, actually do stuff on the directly on the intestinal epithelial cells. So that Daizong then asked, can we actually increase the numbers of the BEST4 cells with a large number, you see some of them here, of these cytokines or chemokines, et cetera. And it turns out that gamma interferon is a very potent activator of increasing BEST4 cell numbers. So this is again the same reporter that you see here. And gamma interferon is known in the gut to be strongly upregulated during bacterial infections. This visualizes the same experiment. So this is a standard organoid, human small intestine. You see in red that there are some BEST4 cells. If we, however, now expose this organoid to gamma interferon, you see a massive increase in BEST4 cells. So we can really manipulate the system now. We can actually create a system where we have many of these functional cells. I should say by single cell sequencing, they are identical to the signature you get out of the primary intestinal epithelium. So then Daizong wanted to create an identical organoid derived from the cell line he was using, but that would be identical in any other way, but would not have BEST4 cells as a negative control. Turns out that when he knocked out the SPIB transcription factor unique to the BEST4 cells, he totally loses the BEST4 cells here. So we have a line that has them, there we can induce more of them with gamma interferon, and we have a control line that's in any other means is the same, but doesn't have best four cells. So then he actually wanted to perform this cystic fibrosis functional test, so the swelling assay I just showed, and he focused on two things. First of all, he focused on the fact that cholera toxin opens the CFTR channel and would lead to swelling. Indeed, I don't show this here, but cholera toxin cannot, when we have no best four cells in our organoid, there is no swelling. And also when we now try to trigger glucat2C, so the receptor for this heat-stable enterotoxin from E. coli, indeed, and I'll show you that we can trigger the receptor here, but if there are no best four cells, the prediction would be that we cannot cause diarrhea. And that's exactly what he sees. So this is the same test that I just showed you in a slightly different version. These are wild-type organoids that do have the best four cells. We add this. You see it here, the heat-stable enterotoxin from E. coli, and there's a rapid swelling. This is actually the loop runs over 1 hour. But we then expose the very same organoid line where SPI-B has been knocked out and there's no best four cells, otherwise are they identical. This swelling assay is totally lost. So how do we interpret all of this? We think that best four cells have a key role in fighting bacterial infections. We showed it for cholera toxin. I show it here for E. coli. What it does, it senses the presence of gram-negatives in particular. It'll then produce a massive outflow of water, and this results in a watery diarrhea, and this presumably then helps to flush out the gut and remove as much of these bacteria, of these pathogenic bacteria, as possible. And on top of that, so when that is not good enough, the gamma interferon that's meanwhile produced by T cells will further increase the best four cell numbers and will further increase the watery diarrhea that's produced in the small intestine of patients. So we think this is at least one of possibly more functions of this novel cell type. And why I show this here, I think it illustrates that, the story illustrates that organoids can help you understand functions of cells that you could not otherwise study in the lab. And there are no cell types, no cell lines, and where there are no animal models, human organoids can come to the rescue. And then my second story, I hope I stay within time, has to do with the liver. Liver has two essential liver cell types, the hepatocytes, the cholangiocytes. Liver is also the most regenerative organ of the human body. Cholangiocytes, or bile duct cells, are very easily grown as organoids. This was shown in my lab by Mary Hoog, actually in 2015, not in 2025. Hepatocytes, very important. I see this now at Roche as well for drug development and for metabolic studies, et cetera, have really resisted long-time culture for a long time. But who in the lab came up with a protocol where we could take a single hepatocyte, put it in culture, and you can see they make this beautiful organoid. This is a mouse. This is now human, very large cells, large nuclei. You see the production of massive amounts of, in this case, alpha-1 antitrypsin, an abundant serum protein. They make albumin. They make all of the cytochromes. And by all means, as far as we can tell, what we are growing here are real hepatocytes. The big difference is that we can actually grow them for many, many months. We have lines now that grow for years. We can subclone them, but they will always look like these beautiful hepatocytes, large nuclei, big nuclei. But you'll see the occasional dividing cell in these organoids. Even more so, they make structures. They make these bile canaliculi. So the hepatocytes produce bile acid and normally secrete it into bile canaliculi. You see that here. We stain for the bile acid transporter MRB2. And it looks like, and it's better to see here, when we grow, so this is a knock-in in the bile acid transporter, green. So we see where the bile canaliculi are. When you have one cell, there is no apical domain, but as soon as an apical domain is formed, the bile acid transporter goes there. And then when you eventually get a real organoid, you can see that every hepatocyte on this apical domain is attached to this bile canaliculus. It secretes massive amounts of bile acids in the canaliculi, and eventually in a real liver, this would flow to a bile duct and would then leave the liver, but we don't have bile ducts in these organoids. The other secreted products like albumin, alpha-1 antitrypsin will actually not secreted to the apical domain, to sort of the bile system, but it will be secreted to the outside, to the basal domain. And you'll find a lot of albumin, for instance, in the medium of these organoids. They can be transplanted. This is now in a mouse liver. The human hepatocytes are red, and actually were done by Ipe de Jong in New York. Actually, from a single hepatocyte, we grew a large batch of hepatocytes. And Ipe transplanted them, and this is a few months after transplantation. And these islets of human hepatocytes keep on growing inside the mouse liver here. Now, finally, can we use this to model human disease, much like I just showed you cancer or bacterial infections? That turns out to be the case. So this disease, steatosis, very common in adult people in the US or in Europe. Up to 25% will have a fatty liver. So steatosis typically doesn't get diagnosed because the real trouble starts later in this process when inflammation occurs, fibrosis occurs, and eventually even liver. So the liver function is lost, and eventually even liver cancer can happen. There are currently no approved drugs. It's extremely common, and of course directly related to the excessive calorie uptake in the Western world, and is closely related to diabetes and obesity and similar diseases. No good in vitro model so far. So Delilah Hendricks set out, this was published very recently, if you want to read up on this. So she set out if you could use these hepatocyte organoids to model the disease. Normally they don't accumulate lipids, they synthesize lipids, but they secrete them in the form of these LDL particles. If we knock out an essential protein in LDL particles, ApoB, it's also a rare genetic human disease. That's what we try to model here. We now see massive amounts of lipid particles. So they do synthesize lipids. There's actually no lipids in the medium, no fatty acids in the medium. They synthesize them from glucose. And then when they can't secrete them, they just simply accumulate them. They, for some reason, don't really care so much, and they happily grow, which is nice for us. We can also model the disease by taking normal hepatocyte organoids and just give them excessive free fatty acids in the medium. So now they basically take it up, synthesize from these free fatty acids triglycerides, and you can see they again get accumulated. So this would be a model for excessive calorie uptake-based development of liver steatosis. There's a lot of genetic studies have been done on MASH, as it's now called, NAFLD. Many risk genes have been found, but often it's not known exactly what the risk gene does. So is a risk allele actually helping or endangering the patient? One of these is one of the most frequently found hit is PNPLA3, a gene of relatively unknown function. So what Delilah did is she took an organoid from a line that did not have the I148M risk-SNP. So it has two wild-type alleles. Then she, by base editing, or by prime editing in this case, she created a single allele or two alleles where the I was replaced by a methionine here, and she created a null. Now, when she created a null allele by disrupting the gene with CRISPR, you can see steatosis results, at least in this in vitro model. And when you create these risk alleles, particularly in the homozygous state, you can also start seeing the modest but clearly present accumulation of lipids. So we would argue that PNPLA3 is a gene you would like to activate to protect the patient. You would not like to block it. Having said that, you can actually find in literature that several pharma companies are currently developing inhibitors of PNPLA3 in the hopes that this would help patients with steatosis. We would argue if you actually inhibit PNPLA3, we do this by a knockout, you actually will get the patient into trouble. We have to see how these trials will come out, and that'll happen, and we'll see that in the next few years. We also got a lot of drugs in development. So as I said, there are no approved drugs, so many attempts. Some aim to block the fibrosis. They will not score in this assay. There's quite a few that, for instance, block triglyceride synthesis. ACC is one, DGAT2 is one. You see some of these drugs work well, others do nothing, at least in these assays. And again, we'll be curious to see if any of these will score in the clinical trials. This is my final slide. So here you see, so from the previous study, we would predict that a combination of DGAT1 inhibitor and DGAT2 inhibitor, these are closely related enzymes that perform a key step in triglyceride synthesis. When the organoids are dark, and I'll play the movie again. So when the organoids are dark, they are full of lipids. You see the droplets here in this phase contrast. So we add the inhibitors over about 3 or 4 days. Apparently, the hepatocytes burn up the lipids and they slowly clear the lipids away from the intracellular spaces. And at the same time, we see that the organoids keep on growing. So our conclusion is from this fairly simple experiment, these two inhibitors, first of all, they help getting a bit of steatosis, of lipid accumulation, but they also are not toxic, at least for the liver, because they allow the livers to happily grow. After we have some other inhibitors that also clear the organoids, but it's clearly not so healthy because they stop growing, and then often in many cases we also see a lot of apoptosis. So to end my second story, what we think is this might represent a model for human steatosis, where there is no good animal model, there's also no good in vitro model so far, which might help actually to come up with treatments for this this very common and also very threatening disease. And with that, I guess I named the people during my talk. I'd like to thank you very much for your attention. I hope I left some time for Q&A. Thank you.
Thank you so much, Hans, Dr. Clevers, for this amazing talk. We have some questions from the audience. I don't know where the mics are over there. Okay, so yeah, the first one is over there.
Great talk. Thanks very much. Jeff Lomax, CIRM. On the gut, the mini gut, what do we call it? The gut organoids, sorry, the first story. I mean, a very exciting opportunity to really build something up in a personalized medicine way, the example you gave. The question really is how close is that, or what's the potential for the automation of that process, and how close are we to standard diagnostics using those tools?
Yeah, well, that is so. I've actually now that I'm a Roche being a very large diagnostics company as well, one of the largest in the world, we've had lots of discussions around this. But let me first say that for cystic fibrosis in Holland and also now some other countries, essentially all patients that did not have access to the Vertex drug, which is about half, because the Vertex drug Orkambi has been in trials and is approved for Delta 508 patients. Homozygous Delta 508 patients, but the other half is a wide array of different mutations. There was no approval for this patient group, so insurance companies typically would not reimburse. And actually, we've made a biobank of all non-Delta 508 patients in Holland, about 600. Virtually all of them have been screened. And we have several dozen patients that actually were found to be responsive in organoids, and black and white, what we hear back from the clinic, they always respond to the drug, and it's reimbursed in Holland. So there's actually now there's a third generation drug from Vertex that no longer makes this very important, because now I think they treat positively about 95% of the patients. But that was done by hand in the lab. Of course, there's not a great hurry in cystic fibrosis, and also the numbers are pretty low. If you think about cancer, so for Holland, we would have 150,000 new cancer patients every year, a country of 70 million people. So there the assay is too expensive, it's too slow, it would take multiple weeks, and also you need highly trained personnel. So there are companies that are with microfluidics in particular try to bring down the volumes and increase the standardization and the reproducibility. I'm actually a co-founder of one of these companies. So Zealis, but I can state that actually it is now possible with these machines, also other companies have set this up in a matter of about 7 to 8 days to go from a small biopsy to a reliable readout on a limited number, say 20, 30 different drug combinations. That's probably what in practice we would need. And this is a machine that basically fits in standard diagnostics lab and can run by a typical diagnostics technician. The big challenge relative to CF is that for CF there was no other drug for these patients, so they either got Orkambi or nothing. So we could immediately, when we predicted something, the doctors were taking the risk to give it to patients, and that validated the assay immediately. Of course, for most cancer types, we just heard examples today. There are many good, if not very good, treatments. You cannot just go in and say, I now have a new diagnostic tool, forget about node pathology or DNA sequencing, this is much better. So this will take a lot of validation, and then reimbursement will be very important. I see how important it is for companies to develop. My sense from what I've seen is actually the reproducibility between labs is high, the predictive value is high. So my sense is there will be space for this, but it might take multiple years before this is embraced by the entire healthcare industry.
Very exciting.
One
Thank
more
you.
question. Kat?
Yeah, as usual, that was a spectacular talk. I don't know if you can see me. Thanks, Hans. I really appreciate it.
I see somebody waving.
That's me.
That's you.
You always see the back of my head at these things, which is better. The best 4 cells, those are very interesting. I thought you were talking about the +4 cell at first, because there was that whole debate. But the best 4 cells being interferon gamma responsive and suggesting that we get immunity to enterotoxigenic E. coli that you showed could actually introduce mutations and initiate malignancy as well. Does that mean if we're using JAK2 inhibitors that also inhibit interferon gamma signaling, we could reduce our gut immunity? Did I interpret that correctly?
Yeah, so the prediction would be that if gamma interferon signaling, we haven't tried it, it's actually a good experiment. If you block gamma interferon signaling, you will not see the increase in these cells, and you will not get this massive diarrhea. Maybe it's also a nice treatment if you don't want to dehydrate patients by their diarrhea. But that would be a prediction,
Okay, great, thanks.
One important component of the gut is the microbiome, and I was wondering how can we combine that with the gut organoids in a productive way? Can you elaborate on that?
Yeah, so there are actually multiple studies already. We try to always work in very simple studies. So Kat was just referring to a study that we published a few years ago, where we asked that a particular strain of E. coli called PKS E. coli, which is quite frequent, about 20% of people carry this particular E. coli in their gut, was known to be genotoxic, secretes a small molecule that interacts with DNA. And we actually, we basically injected them in organoids and showed that they produce a very specific mutational signature. That then was also picked up in cancer patients. So it looks like about 10% of cancer patients have massive numbers of mutations that appear to be caused by this particular E. coli. That's pretty common. So, but that is not the entire microbiome. There was a single E. coli that we could co-culture inside organoids. So there's also labs who are interested in, of course, in the complex microbiome, or at least complex communities that can be propagated in labs. And so there, the main developments have been that organoids, after they have been grown in 3D, they can only essentially be expanded in 3D for long term. But when you have enough of the cells, you can plate them out in a transwell. And there's now some beautiful papers where then in ALI cultures, where you would have an upper compartment and a lower compartment, where the upper compartment would be anoxic. That's where most of the microbiome likes to live. And then the apical, so the microbiome sits on top of the apical domain, and you have access to the basal domain or from the bottom of the transwell. And so this is now, I think, very exciting, because you can now study the effects of the metabolites produced by the bacteria in the gut on the gut epithelium. Vice versa, you can add immune cells below the epithelium and see how that affects the epithelium, but also the microbiome, et cetera, et cetera. This is in development. I must say we don't build these very complex engineered systems, but if you can look them up, they look like they're really going to be very, very useful.
Thank you.
Thank you so much. I think we have we don't have time for questions anymore. I'm sorry. But one went, right? No, it has to be one last one.
One more question, please. Hi,
Okay.
Hans Clevers. This is Carl Willard. I want to see if there's any parallels here between intestinal organoids and liver organoids. You can pinpoint the intestinal stem cell very well, but can you do the same for the liver organoid? Is there a liver stem cell?
No, I think there's consensus in the field now. So my view has become over the years that this classical hematopoietic system with a professional stem cell that is unique, and if you remove it, that's the end of your tissue. Doesn't hold in many other tissues. Like in the gut, we have really dedicated stem cells, but if you kill them, many other cells will take over their role that normally they would never do. So there's a lot more plasticity in solid organs than the hematopoietic system. The liver is the other extreme probably the most regenerative organ of the human body in the mouse body. If a surgeon removes 2/3 of a liver because of colorectal cancer metastasis, for instance, the liver grows back in a matter of weeks. And this is not done by a classical stem cell, what you would call a classical stem cell. It's essentially the hepatocytes that as fully differentiated cells start dividing, which is what we exploit in our organoid culture condition, I would say. So there is a stem cell function in the liver, actually two different ones, but it is not driven by a professional stem cell that you can point at. It's driven by the fully differentiated cells. One is the hepatocyte, I just mentioned, the other is the cholangiocyte, which can actually revert into a stem cell-like state called an oval cell, and then rebuild the liver. And what I would say, I think every organ will have its own strategy to deal with the normal wear and tear and to deal with the sort of pathological loss of tissue.
Thank you so much, Hans, for being with us today and for this outstanding talk. Thank you so much.